Discovering and analyzing the special capabilities of microorganisms could benefit human beings. That's what drives our research forward. A major goal of this Moonshot project is to turn CO2 into a resource and reduce its emission. We aim to develop a process of using microorganisms to fix CO2 with greater efficiency than plants. We're using Ralstonia Eutropha in this project. It's not a commonly found bacteria, but it was originally found in soil, feeding on organic matter. This Ralstonia has a natural ability to make organic matter from CO2, but it takes a long time. So our aim is to speed up this process through genetic manipulation. Microorganisms with the ability to utilize electrical energy have been found before, but our challenge is to induce this capability in other microorganisms through genetic manipulation, which has never been done before. Development will revolve around introducing long gene fragments into cells and identifying genetic sequences that are compatible with Ralstonia. These are the two main issues. Right now, we're aiming to introduce long-chain DNA of around several dozen kilobases. Our ultimate goal is to introduce DNA about 10 times bigger, consisting of several 100,000 bases. Conventional devices can't be used for strands over several dozen kilobases, so we'll need special biotechnology. We're also developing this in our project. First, we need an electrode to supply electricity to the bacteria, so the development of an electrode is extremely important. We're also developing a new gas-phase reaction bioprocess using CO2 as a substrate. We're developing a gas-phase reactor in which the electrode is in direct contact with the gas, bacteria, and water at the same time. The reactor is being developed mainly by a group from Osaka University and Nagoya University. They made a prototype for testing the bacteria we modify. This project is characterized by the involvement of researchers from completely different fields, including those studying microorganisms, catalysts, and electrodes. We're in charge of researching microorganisms. Our role is to introduce the genes required for current consumption, CO2 uptake, and fixing into Ralstonia using genome manipulation technology to simultaneously realize these three functions. Current bioplastics and biofuels are made from food, such as cornstarch and palm oil. If we succeed in putting our technology to practical use, we'll be free of competition for food and turn the problematic CO2 into a resource. That's what we see for the future of society. We're aiming to solve the issues with CO2 and resources by developing a bioprocess to convert CO2 into a resource with greater efficiency than plants.